Good afternoon, ladies and gents. My name is Mrs. Sillier, and today we're going to discuss the topic of DNA and RNA. Most of the time we use different textbooks, so the learners don't always know exactly what they must study. Sometimes they want to summarize the work, but they don't know what is most important and what is not. So your teacher should have the examination guidelines. This is now from 2017, but they will have the latest versions. We should be getting it sometime soon. This should be staying the same. When we look at DNA, for instance, you will see all the information that you need is on this paper, and there's even blocks. When you are studying, you can mark the blocks as you finish, and you will know exactly what is important. Learners always ask us for, ma'am, what is the scope? What must we study? Now, this is your scope. This is what you need to know exactly. Please don't summarize this, because then we will have to summarize your marks. Okay, so ladies and gents, DNA and RNA, let's start, let's look. In order to talk about DNA, we need to know where will we find this DNA, and obviously that will be inside your cell. Just a quick revision of the different parts of the cell that is necessary. So here, let's have a look. This is the nucleus, and here inside the nucleus we have a structure that we call the chromatin network, and inside the chromatin network, that is where we will find the DNA. Around, you will see here we have the nuclear membrane. Obviously it's the nuclear sap that's on the inside, and then here we have the endoplasmic reticulum, and those circle or knobs that you can see there on the endoplasmatic reticulum, that is the ribosomes, which becomes very important when we work with protein synthesis, when we discuss that a little bit later. Okay, so now let's just go a little bit closer to see what is happening inside the nucleus. This is a nicer picture, a close-up. So again, there we have the nucleolus. Outside, there's the chromatin network, the nucleoplasm. There's the nuclear pore where the mRNA will be moving out from the nucleus a little bit later when we explain that. The nuclear membrane, and then obviously on the outside that you can see there, that is the cytoplasm. Okay, just another picture to give you a view from a different side. Again, the nucleus, there we have the ER, and then the ribosomes that is here stuck to the ER. That is why this one is called the rough endoplasmic reticulum. So now at least you know where in the cell we are working, and now we can start discussing the DNA. When we look at DNA, most of us will know that you will find DNA on the chromosome. Now, the chromosomes, when they condense, we see them here in the chromatin. This actually will look for some of you like 2-minute noodles. It is not. This is just a representation of the chromatin network, and when we look a little bit closer, we will see that it's actually where you're going to find the DNA. Here's the DNA double helix, and then it's curled around these molecules that you see here, and we call them histones. They are proteins. Now the DNA is coiled up around this to actually condense, to use less space, because all of this, all the information must actually fit in the nucleus. And as you ladies and gents know, the nucleus are very small, so we need to fit all of that into this small space, and that is why it's curled up like that. All right, now there's different types of DNA that you will discuss, and we will find that obviously the first one, there we have the purple color one, the nuclear DNA. It is called nuclear DNA because that is where you're going to find it. You will find it in the nucleus. Now, the other two types that they mention will be the mitochondrial DNA. Obviously, that you will find in the mitochondria. And then the chloroplast DNA, which you will find in a plant, in the chloroplast. Now, mitochondrial DNA becomes important to us a little bit later when we discuss evolution. I'll explain a little bit more when we look at the functions of DNA. Mostly we're going to work with nuclear DNA because there's more of them, and that is actually where protein synthesis will be starting. Okay. Another picture just to show you that we have DNA is a double helix structure. And then RNA, which we also need to discuss, is a single strand. Now, many times people will say, deoxyribonucleic acid. How am I going to remember to spell that? Now, ladies and gents, when you were in the primary school and you were talking too much, your teacher would have told you many times, now you have to write out the words, don't talk so much, or I will stop talking so much. So you follow the same method when you want to remember how to spell deoxyribonucleic acid. You must just write it out several times until you can spell this. Okay, so this is the DNA. This is the RNA, which is single-stranded, and both of them have building blocks. Now, the building blocks we call nucleotides. Okay, and the nucleotides look like this. You will see this always as the monomer, that is the word that they call it, or the normal word that you know is the building blocks. Let me just try that again. The building blocks. Now, for instance, if you can remember, when we talk about carbohydrates, what is the building blocks of carbohydrates? That will be the simple sugars. When we talk about proteins, what's the building blocks of proteins? That will be the building blocks of proteins will be amino acids. So this is the building block of DNA and RNA, and they are made up out of specific sections. Now, the first one that you can see here is a phosphate. Most of the time when they draw it, the stick figures, they will do it in the shape of a circle. This one represents the sugar. If it's DNA, the sugar will be called deoxyribose. If it's RNA, the sugar will be called ribose. And then obviously there we will have a nitrogenous base. Now these three things, it forms the building blocks or the monomer of all nucleic acids. And the two nucleic acids that we are going to discuss and look at, like I said, it's DNA and RNA. Okay, so for both of them, to build them, what they are made up out of is this. Normally we will say the phosphate and the sugar, that forms the backbone of the DNA or the backbone of the RNA. But it's not always simple, because you will see now the nitrogenous base, there's different options when we look at DNA and when we look at RNA. So let's have a look. This is now a representation of one strand of the DNA. And again, you can see there the circle, that's the phosphate group. Every time in the strand, the phosphate group we will find here, and that is the sugar, deoxyribose. The bond that forms between the phosphate group and the sugar, we will call that the phosphate-sugar bond. There's 4 types of nitrogenous bases that you will find in the DNA: adenine, guanine, cytosine, and thiamine. Now you will already see here that adenine and guanine, their shape is a little bit different from cytosine and thiamine. So let's just have a look a little bit later why that is. Another example to show you the bonds that will form between them. On the previous picture we had just one side of the DNA. Now we have both sides of the DNA. Again, there's the phosphate, the sugar, the nitrogenous base, and that will now be your building block, or the nucleotide, or the monomer. That is most what we call a building block also. So already you can see here that every time we have a thiamine, it combines with adenine. Or here we will see the adenine combines with the thiamine. The same here with the guanine and the cytosine and guanine. We start to see that they are very specific. They do not mix and match. They bond with the same nitrogenous base the whole time. Again, also because of their chemical structure, you can see here, there I have a double bond, there I have a triple bond, but luckily for you, that is not part of the syllabus. You don't have to worry about that. On this side, again, the backbone of the DNA, phosphate, sugar, nitrogenous base. So it's a repetition of the same thing over and over. All right. Now, for those of you who like the science section, let's just have a look why it works like that. I already told you that they pair in a specific way. Now, when we look at the colors here, the cytosine, That is the green one that we see here, cytosine. The chemical structure looks like that. And guanine, the red one, the chemical structure looks like that. Now, these with the double circles, the adenine and the guanine that we see here at the bottom, they are called purines. And those with the single circles, they are called pyrimidimes. Now, as you can see here, cytosine and guanine will combine with one another. And then here at the bottom, adenine and thiamine will combine with one another. That is now when we're busy with DNA. But don't forget, when we work, I'll tell you a little bit later, when we work with RNA, we will not have thiamine. The thiamine is replaced by uracil. And you can see now it's also a single circle. So the shape or the structure will be more or less the same as thiamine, not identical, but that's why it's going to combine with adenine that we have on this side. Terminology becomes very important. You will not understand a lot of the questions if you don't know your terms. So let's just have a look here. Cytosine and guanine, because they combine with one another, we call them complementary base pairs. They are the complement of one another. They complement one another. In other words, they belong together. All right, the same with adenine and thiamine. Now, many times when you look at these sketches that they give you in the exam, you will see they only use the letters C for cytosine, G for guanine, like I showed you here, or A for adenine and T for thiamine. Now, if you struggle to remember things like that, maybe you can remember this little sentence that the car you will find in the garage and the apples you will find in the tree. Maybe that helps you to remember this in the beginning, but later on when you work with this the whole time, you will remember it very easily and quickly. All right. Now, the DNA, because of this phosphate sugar backbone and the nitrogenous bases that we have here, and obviously the weak hydrogen bonds that we're going to have here between them, it will have this specific shape. So that's also one of the questions that they like to ask you. What is the natural shape of DNA? And we call it a double helix shape. If you are lucky enough to have these molecules at school in your class, many of the teachers will build this molecule for you so you can see it in real life, and then you can see that it's a double helix structure. All right, now it always works like this. Somebody didn't just one day decide, okay, this is now how DNA look. We just have to study this. There were people that worked very hard and tried to find out what exactly is this now? How does it look? How does it work? Now that's why we're looking just quickly a brief history of DNA. Now here on the right hand side we have two gentlemen, James Watson and Francis Crick. I included pictures of them that's more recent. So here you can see the last picture here, James Watson, that was taken at the age of 92 and he was still busy working. And Francis Crick unfortunately passed away already, but that gives you an idea. Maybe if we look at the two gentlemen and we see that they are losing hair at the top, maybe that is because they have to work so hard with DNA. We'll have to check your grade 12s. Maybe something is going to happen to you as well when you work so hard the whole time. Okay, so Watson and Crick, they made a discovery. They were given the Nobel Prize for discovering the shape of the double helix. But it wasn't always that easy. While these two gentlemen was working at Cambridge University, there were two other people also working and finding out stuff regarding DNA. That will be Maurice Wilkins and Rosalind Franklin. They were working at King's College in London. Wilkins and Franklin, they did not get along very well. So he was actually a little bit irritated by this woman. So he decided, when she worked with the X-rays and she saw this specific diffraction pattern of DNA that we have here at the bottom, when she found that and she saw that, Maurice Wilkins decided, you know what, I'm going to tell Watson and Crick, because they also work with this, I'm going to give them the information. Now, you may not know this, but universities normally they compete. These scientists compete when they do research because they want to be the first one to find out the new information. And obviously when they find out stuff, they have to publish this in scientific magazines. And if they do well, then the university will get sponsors and money, and that will just keep them going. So actually there's a rivalry between them. It's not a good thing when somebody tells somebody else what is happening, and then they will get all the fame and the glory. So unfortunately, Rosalind Franklin at that stage, she was sick, although she was still very young. She passed away because of cancer. And then here in about 1960 something, because all of this happened now in the 1950s, which is a very long time ago. So in the 1960s, what happened is they received the Nobel Prize, Watson, Crick and Wilkins. They did not acknowledge the fact that Rosalind Franklin also had sort of a contribution here, but luckily today I see lately in the news they do recognize her, and some of the universities even have a plaque, or one university I saw was named after Rosalind Franklin. So at long last, they also acknowledge the fact that she actually, if you ask me, she gave the information that is needed to find out. So before you get stressed, all you need to know about the history of DNA is that Watson and Crick were the guys that found out and received the Nobel Prize for the double helix shape, and that there are complementary bases, that A combined with T and C combined with G. So that is the history. All right. Now, the functions of DNA, just quick, we will discuss this in full a little bit later. The first function, which is very important, DNA we find in the cell, and the DNA is going to be the template to make the protein. So all the proteins that you have in your body, it is made starting with the DNA. So we have enzymes, that is proteins, we have hormones, that is proteins, we have muscle tissue. So all of those things must be made, and we need this process, protein synthesis. And remember, synthesis means to make something, to make proteins. So the first function of DNA is to make proteins. There's a nice picture showing this. We will look at it closely a little bit later. And then secondly, your hereditary characteristics, the fact that you look, like your mom or your dad or maybe your grandfather, it is because of your DNA, and that is your hereditary characteristics. A little bit earlier we talked about mitochondrial DNA, and the mitochondrial DNA will be found in the sperm cell and in the egg cell. But the moment, let me just show you quickly here, when we have the egg cell here and the sperm cell there with the nucleus, when fertilization takes place and the sperm cell enters the egg cell here, that section where we will find the mitochondrial DNA, it is discarded. It's only the nucleus that's going into the egg cell. And then so you, as a boy or a girl, you will only have the mitochondrial DNA of your mother. So when you look at this picture, for instance, it shows you that mitochondrial DNA can give you the female line of your ancestors. Mitochondrial DNA will give you the female line, because mitochondrial DNA as well, they are a lot smaller than nuclear DNA, and mutations can happen much faster. So it's easier for us to see the markers of mutations on the mitochondrial DNA. So we just look for those markers, and then because of that, we can trace your female line or your ancestors there. Then normally when we look at the nuclear DNA, it gives us information of all the people that came before you. So actually you are a copy, you are a walking copy of all your ancestors. So you're supposed to make them proud because you carry a little bit of them in your cells with you. And obviously you're going to transfer that to your children one day when you have children, because all of this is now in your DNA. All right. Now, ladies and gents, this is now the first process that becomes very important for us, DNA replication. Okay, so now what happens is you get injured or you have a cut, and then the cells need to be replaced. So if you have a cut on your skin, you don't want other cells to replace it, you want skin cells to replace it. And that is why this process, DNA replication, becomes so important. That's the first process that we need to look at. Now, let's have a look. Why is this happening? Why do we have DNA replication? We need new cells to form that will have exactly the same amount of chromosomes as the original cell. Now, I'm sure you guys and girls know by now that our cells all contain a diploid number, a double set of chromosomes, which is 46 chromosomes. So now if I want to make another body cell, which is a somatic cell, I need again 46 chromosomes. So I can't now go and take a body cell with 46, divide it by 2 and say, Bob's your uncle, there you go, now you have another cell, because then it won't have the correct amount of chromosomes. We need those numbers to double first, which is going to be 92, and then it can divide again to give us 46 and 46. So that is why DNA replication is happening. Here they say it very nicely. So they say it doubles the genetic material so it can be shared, and then when we have the daughter cells, they are identical. All right? This is what they say here, identical daughter cells. So we have exactly the same. The next question that they will ask you about will be, where is this happening? Now, we've been talking about this the whole time. The DNA we find in the nucleus, it can't leave the nucleus. It's going to stay there the whole time. So where will we find this process happening? In the cell's nucleus. Okay, next question. Let's change colors a little bit. When will this happen? Now, ladies and gents, some of you may already be busy or may have finished the process of meiosis. Interphase will happen before meiosis to make gametes, or the grade 10 work where we did mitosis, you will know about interphase, where we have DNA replication. So interphase happens before mitosis and before meiosis, and then that is when we're going to have this process of DNA replication. Now the big thing is, how is this happening? If we are lucky enough, they can ask us to describe the process of DNA replication, but it's not always that easy. So in order for you to apply this knowledge, you have to study this, you have to know this. There is no summarizing this or a shortcut to make it easier. This is what you need to study. So let's have a look how it works. Here's a picture, a representation of a DNA molecule. Now, I'm going to do this so that you can see. There's our double strand. That is our phosphate sugar backbone that we have there. And obviously, if it wasn't for the fact that we want DNA replication, it carries on all the way. It goes all the way like that. Okay. Now, what happens? I have here the DNA double strand, then I have the nitrogenous bases there in between, cytosine, guanine, adenine, thymine, and now because of DNA replication, let me just add that, it's not here, the DNA double helix will unwind. This is our DNA double helix. There it shows us, it will start to unwind. It will not be twisted anymore again. It's going to be flatter like a ladder structure. It looks like this, okay? So when it looks like this, the next step is going to happen. That weak hydrogen bond that we have there, that weak hydrogen bond is going to break, and then this DNA double strand is going to unzip like a zipper. Okay, here they show you weak hydrogen bonds between the nitrogenous bases break and the two DNA strands unzip. Okay, now they are open. There's now possibilities for other nitrogenous bases to come and combine with them. But remember now, what did we say? They are very specific. Guanine will only bind with cytosine and thymine will only bind with adenine. Okay, so here we see the word template, there we see the word template. So each one of these original DNA strands, they are going to serve as templates for these other strands to form. Okay, they are going to be the guide, they are going to be the recipe. So around the DNA strand here inside the nucleus where we are, there will be free-floating nitrogenous bases or nucleotides with their phosphate and their sugar. And they will come and combine in a specific way. Here you can see already. Here's the adenine coming for the thymine, the same on that side. Here we have a thymine coming from the adenine, and here there's a cytosine for the guanine. So they're going to start to combine, and they're going to form weak hydrogen bonds here again. And that is why they say here at number 4 that we're going to have complementary strands forming. Have a look here. Free nucleotides to build a DNA strand using the original strand so that A and T, C and G will combine with one another. So that is how you explain it. And then here at the end, what do we have here? We have 2 identical DNA molecules, because remember, DNA is a molecule. We have identical molecules, and then the replication took place. So the same strand that we have in the beginning, now we have a copy of it. That's why they say 2 identical DNA molecules. So that is your DNA replication. You study it exactly like this. If we're lucky enough to ask it. Otherwise, they will ask you questions about why, where, when, and then sometimes they give you pictures and ask you to identify the process and label certain sections and describe certain things. So make sure you know exactly what is happening here. All right. Now, ladies and gents, we have DNA. So DNA, we should be able to use it to our benefit as well. So here you can see I have a picture that they talk about a DNA profile. Now, every single person today, you have DNA, so you will have a DNA profile. And some clever people a long time ago decided to now use this. And that is what we use today in at crime scenes, forensic science, and then also when we want to determine paternity, if somebody is the parent. So we use DNA profiling. Here you can see a nice picture where the scientist is showing us a DNA profile, how it looks on the X-ray film. And then this guy that we see here at the bottom, Alex Jeffries, he's actually the guy that discovered this specific technology, and then we can use it, to identify siblings, or then obviously if it's a crime scene, to try and catch the perpetrator. All right, now the first thing that you need to know is what is a DNA profile. So you need a definition. Now when we look at that as well, we can see it's a pattern or an arrangement. So the word they like to use, it's a pattern of black bars or an arrangement of black bars. Don't worry so much about the X-ray film, but this is a pattern or an arrangement of black bars according to somebody's DNA, And when we have this pattern, we can now compare this pattern with another known profile, which they give to us here. So we get some blood from a crime scene, or we get saliva, and then we can compare it with suspect's saliva, and then we will know, okay, this may be the guy, and then they can use that to build their case. Okay. Now, here's just some more pictures to show you what is DNA profiling used for. Here's a picture of sickle cell anemia that you also will discuss later. That is when your red blood cells have the shape of a sickle. The surface area is much smaller, so the amount of oxygen that can be carried will be less, and that will have some consequences for your health. But if we can now use the DNA profile to determine inherited disorders and identify them, obviously we can now use that as well to develop cures and do research. So DNA profiling is helpful for people that's doing research as well. Then thirdly, it can be used as biological evidence. I said to you now, these guys are now twins, so we will find that their profile will be similar. They will have the same DNA, but we can also use it in identification of relatives. In other words, we find a body and people don't know, they need to identify this body, then they can use the DNA profiling method. And then obviously, like we spoke about now, we use it a lot for evidence. Paternity testing also becomes important because they do blood tests, but when you do a blood test, then you can only actually say who may possibly be the father. You still need to do DNA tests to confirm that. All right, now there's just another nice picture showing you how it looks when scientists work with DNA. This picture is not as clear, but it's just the process. It shows you have a blood sample with your blood in it. Then they extract the DNA. They cut certain sections using enzymes. In other words, they use that section that is different. They say that 99% of our DNA is the same, so that 1% or 99.9%, that 0.1% that is different. That is what they use to do this. It goes through a process of gel electrophoresis, and because of polarity in the DNA, it gives us that pattern, and then that is the pattern that they use that you have seen now on the previous pictures for DNA profiling. Now, always when there's something that can help us, there's always problems or disadvantages when it comes to this. Many people will say that the DNA that we use is a small piece. So what happens now is that could be similar. If we're only using a small piece, that piece can be similar to the piece of the DNA of another guy, so that becomes a problem. And then secondly, humans, people are doing the testing in the laboratories. So let's say today you're not feeling so well, you are tired or sick, or you have problems and you don't focus, so you may contaminate the sample, and then it's going to influence the standards and the controls, and they can use that in court against you. Then thirdly, DNA profiling, the moment I feel you work with anything concerning medical doctors or tests, it becomes expensive. So people will say this is very expensive, so they don't want this to be used. And then lastly, if they have your DNA, they will have your information, and now they can use it against you. Some people don't want any of their information to be used, and then they appeal against this. They are not happy, or they don't feel safe or calm about this being used. So sometimes when they ask you to discuss the debates, remember to stick to the theory and then just explain with facts why there are debates regarding the DNA profiling. All right, now here's just a few examples and more pictures. If you like me, you like pictures in color, and I study that way, so it will help you to remember as well. The different samples that we can use. Here's examples: blood, hair, saliva, semen, body tissue, vaginal cells that's transferred. All of those things contains DNA, and that is what we use to make a DNA profile. Okay, now here's just an example of a case study. A violent murder occurred. The forensics team retrieved a blood sample from the crime scene, and then they prepared this profile now, and it looks as follows. So let's have a look. Was the suspect at the crime scene? Okay, now these bars, this is now your DNA profile that you work with. Here is the suspect's profile. That is the victim, so obviously it's just to make sure if we find some evidence that we can exclude the victim. And this is the blood sample from the crime scene. Now immediately when you look here, you can compare it and you see, but these lines, they are the same, they match. So this suspect's profile matches with the blood sample that we found on the crime scene. So now they can actually use this as evidence in the court. They still have to, gather enough evidence to link these things, but this is a start. This can help them to try and find out and catch the criminals in certain situations. All right. Then another one that I want to look at is the cases of paternity. If somebody is the parents and, can they now start paying for the child that's also theirs, and they are responsible, so they have to do their part. Important to remember here, when it comes to the DNA and the profile of the father and the mother and the father's DNA profile the child's will be a combination of the two. So you will see the man, it's not exactly the same as the child, but there's places where it overlaps, where it is the same. And then again, you will see the mother as well. So the mother, she that is the same here, and the fathers, those are the same. So the child is a combination of the two. So don't let them trick you if they maybe give you one like this and now you think, oh, but it's not 100% the same. It will not be, but there will be a lot of overlapping taking place, and that shows us that this man is actually the father of this child. This is how they use it. Just a few famous cases. The actress Elizabeth Hurley, she sued this guy Stephen Bing, said he's the father of a child. So in the beginning he said, no, it wasn't me. So she used DNA profiling to determine that he was actually the father of a child. Another interesting case I just want to show you, this one of Colin Pitchfork. That was the first guy that were actually convicted because of DNA profiling. If you can remember when we looked at that picture of the guy that actually, I want to say, invented DNA profiling, that was around in 1985. And this guy committed crimes here in 1986 and 1988. And this was the first case that they actually used DNA to convict someone. Okay, another famous case that you may know of is the case of O.J. Simpson, and this is also the case where they showed that DNA evidence can also have a negative side where it can be contaminated or they will not accept it because of several situations that we discussed previously. All right. Another picture, just to try again to make sure you understand. Crime scene. Suspect 1, suspect 2, suspect 3. All right. And immediately you can see already that these bars, they match. All of the bars in the sample for suspect 2 match with all of the bars in the sample for the crime scene. So obviously then suspect 2 will be your guilty party. A little bit later we're going to practice a few questions. Then you will also see how this comes into play. Okay, so that is DNA profiling. We have discussed DNA now over and over, so now we have to look at RNA. Now, many times in the class, the learners say RNA is the player in this relationship because RNA is a single strand. There's no bonding with another strand here like with DNA. It is single. It will have no weak hydrogen bonds. It's going to be a shorter molecule. And then obviously there's one big difference from DNA, and that is that thiamine is not here. We have uracil. So when we work with RNA, we're going to have a different nitrogenous base, which is uracil. The structure of the RNA is still the same. Remember, we will have our phosphate, the sugar, which will now be ribose, and then the nitrogenous bases, which will be cytosine, guanine, adenine, and uracil. So they like to ask you what is the differences between DNA and RNA. We will also look at that. Sometimes they ask you to tabulate that, and then it's easy. Then you just write it down because you just studied the differences. Okay, now where are we going to find RNA? RNA will be inside the nucleus and outside of the nucleus because it's a single strand. It can move. Like we said now, RNA is a player. He moves around. He does not stay in one place. Okay, so we have 3 different types of RNA, which becomes very important when we look at protein synthesis a little bit later, the making of proteins. So this first one that's going to start building the message, we call the messenger RNA, and in short, normally when we write about this, we will say mRNA, that is the messenger RNA. Then the next type of RNA that we have is the transfer RNA. It's going to transfer something. You see there is something at the top. That will be the amino acid. That amino acid will be transferred in order to make a protein. That is why this is the tRNA. And then the other type of RNA that we get is the ribosomal RNA. It's called rRNA, and obviously you find that at the ribosome in the cytoplasm. All right, now let's have a look. The differences between DNA and RNA. Nicely colored picture. Okay, First of all, when you look at the picture, you can see it immediately. The DNA is double-stranded and the RNA is single-stranded. Normally, that's the first thing that learners mentioned. Then, the other big difference, we have uracil when it comes to RNA, and then we have thiamine when we work with DNA. Then RNA will be a shorter molecule where DNA can be a very long molecule. Then also the RNA will have no weak hydrogen bonds, but the DNA will have weak hydrogen bonds. Okay, so maybe you like colors, so you will remember this. This is also part of a parcel that we received from the department. It helps a lot when they talk about stick figures. This is like a stick figure. They give you the DNA on this side, the RNA on that side, all the different sections that we have discussed now, the building blocks of DNA. There we have the building block of RNA, phosphate, sugar, nitrogenous base. We have the weak hydrogen bond, and there we have no bond. So that is normally what they ask you to now compare DNA with RNA. And then remember, when you are comparing information, ladies and gents, if you write a fact there, the fact that you want to compare with that one, you can't now write it at number 3. You must write it on the same line and compare it with the same thing. Many times learners swap this around and then you lose marks because you are not comparing the different factors. You are just listing them in a block formation. Then you cannot get the marks. Remember that. Okay. All right, now we get to the nice section, protein synthesis, the making of proteins in the body. First I'm going to tell you the story, then we will look at the facts. Okay, when we work with protein synthesis, there's two spots where this happens. You need to get your GPS working. So the first one, this big green structure here, is the nucleus. So something is happening in the nucleus, and after that, something is going to happen at the ribosome. This one, the children always tell me it looks like a butternut. This butternut, this structure, is a ribosome. Okay, so the two processes of protein synthesis, the one will happen inside the nucleus, the other one is going to happen outside here at the ribosome. Okay, now which nucleic acid do we find in the nucleus? We said that already. The DNA cannot move, it stays there. It is only the RNA, look here, the single strand. This single strand can move out of the nucleus because there's nuclear pores, there's an opening. So this mRNA strand that we see here is exactly the same. It is actually that strand. It's just to show you that this specific strand is now moving out. It's going to attach itself here to the ribosome here, and then it waits for the tRNA to bring the amino acids. So this process that you see here, it starts in the nucleus. The messenger RNA that's carrying the message is going to go out through the nuclear pore. It's going to go to the ribosome and wait there for the tRNA to bring the specific amino acid. And a few of them are going to combine in a specific way, a few of the tRNA molecules, and then it's going to form a strand, and that strand is going to curl up and become a protein. So if you have the story, now we can look at the specifics, because when you answer this question, you need to be specific. Let's have a look. Transcription. Transcription is the first process. Transcription is now the process that is happening here in the nucleus. Okay, so this DNA strand is going to unzip. It's going to unwind first. It's going to unzip. The weak hydrogen bonds are going to break, and then the free nucleotides that are here, they are going to combine. Something to remember when they talk about script. Sometimes people will say, read from scripture. In other words, you have to read the message in a book. A script is a book. Something is written. So that is what's happening in the nucleus, transcription. They are writing a message. Now, in the beginning, I showed you the guidelines. And in the guidelines, they have a full description of how transcription and translation must be answered. This is just a copy of what is in your guidelines. And that you must study. You must copy. You must be like a copycat. You must study every single word and give it when they ask you. If we're lucky and they say, describe transcription, this is what you write. Again, I showed you script, meaning the writing of the message. That is where the message is written. These pictures show you again, this is the DNA strand. It is now inside the nucleus. So first of all, this double DNA helix will unwind. It opens up. This opening up makes that the weak hydrogen bonds will break because this strand and this strand now unzip. Then the next thing is the free nucleotides that's floating around in the nucleus. They are now coming and they are going to combine according to nitrogenous bases. Now you need to remember when we work with DNA, we have thiamine. But the moment we work with RNA, we don't have thiamine anymore, we have uracil. So this is our DNA template. We call it our DNA template. And then 3 of these nitrogenous bases next to one another on the DNA template, we call our DNA base triplets, like 3 babies, triplets, okay? And they will code for the nitrogenous bases that we have on this mRNA. This is now the mRNA. And because it's RNA, when I start with an adenine, if that is an adenine, it is not a thiamine that's going to combine with that mRNA, with the DNA. The mRNA will be a uracil. The next one that is thiamine here, thiamine will combine with the adenine because mRNA will have adenine. Adenine is still part of the nitrogenous bases of RNA. And then that one, for instance, will be UAU. So that message is the way that the mRNA codes itself. It makes a mRNA strand, and then from there onwards, it's now going to move. So this whole section, transcription, how they code the message, how it is written in the nucleus, is according to the DNA template. That is how the mRNA is formed. The first, or the bases, the triplets next to one another, we call a DNA base triplet. And then on the mRNA, the triplets we call a codon. So the codon is going to combine with the base triplet, or it's going to be formed according to the base triplet. And when you study this transcription, this is what you write down, and you will find it in your guidelines, obviously also in your textbook. In your textbook, it may be explained a little bit, with longer sentences and information that you don't need to know. So this is exactly what you need to write when you describe transcription. Now we looked at transcription now. Transcription happened now inside the DNA. This mRNA strand are now moving out. It's going to lie on the ribosome. And the moment we're at the ribosome, now we are busy with the process that we call translation. The message is there. The message is on the ribosome. But now we need a molecule to translate this message, to bring the amino acid that we need. And that is where the tRNA molecule comes into play. You can see here they talk about a clover shape, the specific shape of the tRNA molecule. And it's going to bring an amino acid. And specifically here it has SER. It's a specific amino acid. And obviously we will have specific coding there that codes for that amino acid. Now in translation, what happens now? There we have the mRNA strand, the 3 nitrogenous bases together we call a codon, and then the 3 nitrogenous bases of the tRNA that's going to combine with a specific group of codons on the mRNA we call anticodon. So the anticodon is on the tRNA. Anti means most now opposite, against. So it is the codon against the mRNA codon, opposite of the mRNA. So that will be the anticodon. And then the tRNA brings the amino acid that is there. According to that sequence, many different tRNAs will come. You will see there's already an amino acid. So a tRNA brought that amino acid but left already. So we are in the process to see the next one coming here, and obviously it will carry on like that, and all of them will combine together to form a protein or a protein, and then here in between them there's a bond. Every time there's a bond forming, and we call that bond a peptide bond. So they attach themselves with a bond called a peptide bond. And when the sequence is finished, also the amino acid sequence will be finished, and we will have our protein. So it is important to remember when you describe this, you have to specify and say each tRNA carries a specific amino acid. And then the anticodon, you can't just say the tRNA, the anticodon on the tRNA will match or combine with the codon on the mRNA, and then they will say the tRNA brings the required amino acid where? To the ribosome in a specific sequence. And then these amino acids form a peptide bond, and that's going to form your protein. So, ladies and gents, this is the theory according to the guidelines that you need to know. I say again, you can't summarize this. This is what you need to know to do well. If you need any information, I'm sure you can ask your teacher. They have the guidelines. They have all of this. If they haven't given it to you yet, I'm sure they will now. So good luck, and I hope this helps and you understand.